Okay, thank you for that very kind introduction. And then again, also special thanks to the seminar committee for the invitation to present today. As mentioned, my name is Carl Manos, and I'm currently a postdoctoral scholar in Kalen Plesch's lab at the University of Oregon. And today I'm excited to share how we build large-scale synthetic gene libraries and use them to decode variant effects in antibiotic resistance. Let me just change my view here real quick. Okay. To start, I want to do just a quick disclosure. As mentioned, I'm also a principal scientist at Simplexity. And so this is a biotech startup company that's been founded out of the Plesha Lab at the University of Oregon. And so speaking of the Plesha Lab, we specialize in developing high throughput gene synthesis technology to design and characterize protein families through functional assays. And so we start with sort of that classic synthetic biology feedback loop of design, build, test, and learn. And for the design aspects, we focus on designing large data sets of protein variants that we mine from protein databases. We get them from metagenomic data sets, engineered variants, and then more recently, we're working towards machine learning generated data sets. And so once we've designed our different protein libraries, we build them using it's a proprietary dropsynth gene synthesis platform from our lab. And so with dropsynth, we can build up to over 1,500 gene variants per reaction. And then we're currently prototyping a 12K version soon. So that'll increase our output by an order of magnitude. And so our dropsynth process, it's capable of generating gene variants of up to 1 kb in length. And again, we're also optimizing to process up to 2 kb gene lengths in the future. And so at a cost of about 70 cents per kilobase, our gene synthesis platform is capable of generating these large synthetic gene libraries at a really incredibly affordable scale. And so then after we build our dropsynth libraries, we test them in these multiplex functional assays. And so we transform these scalable variants again from hundreds up to thousands of gene variants into knockout models, and we quantify their fitness effects or their response based on their growth and then recovery under different experimental conditions. And because each of our gene variants is assembled and then tagged with a unique 20 base pair barcode, all we have to do at the end of the growth experiments is just recover the barcodes from the surviving cells, and then we can use NGS like NovaSeq to sequence billions of barcode reads to do our fitness calculations. And then finally, after testing our synthetic genes, we can learn a lot about the sequence function relationships using high dimensional mapping and novel analysis methods that are designed to handle these diverse data sets. And so we're also moving towards developing predictive models from the gene fitness data that we can feed into machine learning models to hopefully gain even deeper insights into our highly diverse data sets. And so for today's talk, I'm going to be focusing on how we use dropsynth to explore how divergent evolution can shape antibiotic resistance across an entire protein family. And so our goal was to define which mutational adaptations enable resistance and then how those changes can vary across related enzymes. And so as just a quick recap, right, we know that antibiotic resistance often arises when a few of these resistant variants survive maybe a broad spectrum treatment and then expand once the susceptible populations are eliminated. Yet, since most microbes can't be cultured in the lab, it's often really difficult to directly measure how mutations affect fitness or drug response. And so by synthetically reconstructing and now testing thousands of these different gene variants simultaneously, we can start to sort of systematically map how sequence divergence can influence resistance, hopefully revealing patterns that traditional, maybe culture-based methods can't quite capture. And so that brings me to our proof of concept study. This is where we applied our dropsynth approach to decode antibiotic resistance mechanisms within the dihydrofolate reductase, or DHFR protein family, a classic target of antifolate drugs. And so DHFR is an essential enzyme in the pyrimidine biosynthesis pathway, catalyzes the reduction of dihydrofolate to tetrahydrofolate, and it uses NADPH as a cofactor. And so it's also a key antibiotic target because it's inhibited by trimethoprim. This is a synthetic antifolate that blocks the NADPH binding pocket, and then in turn it disrupts DNA synthesis. And in our case, DHFR offers an ideal model system, right? It's well studied, it's clinically relevant, it includes thousands of naturally occurring variants, and so that provides a really exceptionally rich data set for designing our synthetic gene libraries. Now, for the actual drop synth assembly, I was working with Kalen Plesha, along with a former undergraduate in the lab, Carmen Resnick, and our lab manager, Sam Hitton. And so DropSynth, it starts with the microarray synthesis of oligonucleotides, representing fragments of, in this case, a 1536 design DHFR library. We then use oligo-nicking enzymes, and we expose a single strand end of each oligo, and that exposes a unique barcode that's designed to anneal to a corresponding barcode attached to the microbeads. And so these microbeads, or these barcoded microbeads and matching oligos, are then selectively pulled down into these water and oil droplets. And these are the fundamental components of our DropSynth assembly. They undergo emulsion PCA to assemble the full-length genes. And so now here's an actual microscopy image of our droplet pool, and this is containing the microbeads. And so each droplet, you can see, contains a single microbead, and it'll have its matching oligos that are needed to assemble a single gene. And so it's this compartmentalization that removes this potential for cross-hybridization between the oligo fragments for different genes. This allows us to have this proper full-length assembly for the large number of genes in the same pooled reaction. And then after breaking the emulsions, we can recover all of these assembled genes. They're then barcoded with another unique, that 20 base pair barcode, ligated into an expression plasmid, and then transformed, in our case, into DH5 alpha cells. And so by the end of this process, we've actually generated the most phylogenetically diverse library of synthetic DHFR homologs to date. So this represents over 780 species of bacteria across 17 different phyla. It includes many clinically relevant pathogenic variants that are known to play a significant role in antibiotic resistance. And then to explore the actual antibiotic resistance mechanisms, we couple our gene synthesis approach from DropSynth with what we call broad mutational scanning. And so within the DHFR library, when it's assembled and transformed into these DH5 alpha cells, this helps to increase our yield. We then extract the plasmids and we transform them into an E. coli knockout strain, what we call ER2566. And so this knockout strain is missing the full A gene for folate biosynthesis. It's also missing the Thi-A gene, and we've actually built that into our expression plasmid. It helps to prevent metabolic toxicity effects that are known to be associated with DHFR overexpression. And then from here, we grow the cells in minimal M9 media, and then we evaluate their ability to rescue metabolic function without supplementation. This is what we term complementation. And then we also test their ability to resist antibiotic inhibition across a trimethoprim gradient. It ranges from low doses, including the minimal inhibitory concentration, up to 400 times the MIC at 200 micrograms per milliliter of trimethoprim. And then following growth across our experimental gradient, we can actually recover the cells and then sequence their barcodes for surviving homologs and mutants. And then we assign fitness scores to the sequencing data based on the recovery counts. And we use our broad mutational scanning approach to evaluate complementation and also the resistance metrics. For complementation, we were assessing the DHFR homologs for their functional complementation again in these E. coli knockout strains. And so in this first figure, you'll see we successfully assembled 1,208 unique DHFR homologs across 2 codon versions from our original 1536 designs, of which we then actually only recovered 1,136 homologs from our transformation. So that represents about 94% of the assembled library successfully was transformed into the cells. And then once we've calculated and assigned fitness scores based on this barcode recovery from the transformation assays, we can set sort of an arbitrary fitness cutoff. In this case, anything greater than -1 indicates if the homolog was capable of complementing function in our growth media. And so you can see now in this case, we found that about 50% of the assembled homologs had fitness greater than -1 and could therefore complement function in the knockout strain. However, as part of the DropSynth assembly, we also generate a collection of mutant variants that are associated with each of the designed homologs. And so these mutants, they occur for a variety of different reasons. They could include errors in the oligosynthesis, inefficient gene assembly within the droplets, and then assembly mistakes during PCA. However, we can take advantage of these mutant variants and evaluate the genetic flexibility of these different individual homologs when incorporating amino acid substitutions. And so here you can see basically that when we incorporate mutant variants with only a single amino acid substitution from the designed homologs. So basically, at point 0 on the x-axis, that's what I'm showing in the first graph. That's the 50% that can complement. When you add just a single amino acid substitution, you go from 50% being able to complement all the way up to 87% of the unique homologs being able to complement. Right. And then when you incorporate mutants with up to 5 amino acid substitutions, we can recover over 90% of the homologs that are capable now of complementation. And so these mutant variants, they really greatly expand our usable data set, and they help fill in the sequencing gaps that you would probably usually overcome with more traditional methods, such as deep mutational scanning. So now here's a look at the fitness correlations between these different codon-optimized libraries, where the majority of the shared homologs are capable of complementation in both codon libraries. However, you still have this subset of homologs where complementation is pretty codon specific. You can see the points in blue correspond to the codon 1 library, and then the points in orange at the top correspond to the codon 2 library. And then, of course, there's also a set of homologs that were unable to complement function in either codon library. And from this data, we classify our recovered homologs by their fitness response as either complementing homologs, where their fitness was greater than -1, or as dropout homologs with fitness less than -1. And from here, you can see that this sort of binary fitness cutoff is distributed across the phylogenetic range of our DHFR homologs. There's some clades consistently capable of complementation, and then others clearly not. And so I'll start first with the dropout homologs. We use these for evaluating the gain of function mutations for the recovery of metabolic function. And we're only focusing on single amino acid substitutions that were able to recover metabolic function from the data set. And so what we do is we align these gain of function mutants to identify positions along the protein sequence where specific mutations were significantly enriched among the functional recoveries. And so from this analysis, we're actually able to identify, in this case, 7 residues that showed this statistically significant enrichment of gain-of-function mutations. And so these indicate key sites that are probably contributing to the recovery of the enzyme function in the E. coli knockout model. And then from here, we can actually map these gain-of-function positions onto the wild-type E. coli DHFR structure and then visualize where these mutations exert the strongest effects on fitness. And so again, from these 7 different gain-of-function sites, 6 of which occurred on this alpha helix, and it directly interacts with the NADPH molecule. So that helps to stabilize its position within the binding pocket. And so mutations at these sites, of course, had the largest impact on restoring catalytic function, highlighting their sort of central role in fitness recovery. Now, on the other side, we can look at the complementing homologs. These have fitness scores that were greater than -1. And we use these now in what we call broad mutational scanning. And so in this approach, what we're doing is we're quantifying the fitness effects of every possible amino acid substitution at every position across the complementing homologs and their associated mutants. And then we collapse these fitness values into a consensus profile, aligned by position along the protein sequence. And then that helps to reveal how sequence variation shapes functional tolerance across the protein family. And so we end up with a heat map showing the fitness response for each of these amino acids at each position across the sequence under these complementation conditions. And so on this heat map, the black boxes represent the E. coli residue at each position. And then what you might be able to sort of quickly deduce is that nearly all of these amino acid substitutions recovered from the complementing homologs infer sufficient fitness for complementation. Right? There's only a few instances across the heat map where a specific amino acid substitution appears to have a negative fitness effect, shown more in those dark purple squares. Right? And so this is to be expected since we only incorporated fitness responses from those homologs capable of complementation. Right? So this is our baseline for now evaluating the fitness responses to antibiotic treatment across the trimethoprim gradient. Okay, so now from here, we're going to be evaluating the DHFR homolog fitness across that trimethoprim gradient. And so what we start with, with the complementing homologs, we see that as our baseline and measure their fitness response. And then as expected, the fitness in both codon versions starts to decline as the trimethoprim level increases. Right? And then where it starts to become a little bit more interesting is when we look at these low distance amino acid mutations. So the first graph here showed just the design variants with 0 amino acid mutations. And now I'm showing with one amino acid mutation how fitness is starting to change. And then we can go out to 5 amino acid mutations. And you can start to see, especially at the highest end of the trimethoprim gradient, that you're starting to get very codon-specific responses with these different mutations, where you have a lot longer tail of resistant variants, especially in codon version 1. But when we compare these sort of two codon libraries, we can see again now at the minimal inhibitory concentration, the clinically relevant levels, right? Most homologs still remain capable of complementing function, right? And so this is even under that selective pressure strong enough to usually inhibit pathogenic variants in the clinical setting. However, when we then switch over to that 400 times the MIC, the far end of the trimethoprim gradient, right? The upper end of our, the majority of our homologs start to lose function. And we're showing very low fitness values across both codon versions. And notably, of course, there's a few high fitness homologs that persist. Some are shared between the codon versions, some are unique to one version or the other. And then these indicate exceptional resistance to trimethoprim inhibition. Okay, so now returning back to the broad mutational scanning analysis, we map the fitness response across these protein family by collapsing all of that homolog data onto the amino acid specific effects along the protein sequence. And so at the MIC shown in the heat map on the top, we observe numerous substitutions that produce a negative fitness response, right? These are reflecting mutations that disrupt function under mild selective pressure. But now when we look at that 400 times MIC, the heat map on the bottom, we have the perspective sort of shifts. So most residues now show strongly negative fitness, and our focus now turns to identifying the rare substitutions that actually confer a positive fitness response, right? We're trying to reveal the specific mutations that enable survival under these extreme antibiotic stress. And so what can we do with sort of the insights generated from our BMS analysis is that we can map these average fitness responses for each amino acid position onto the wild-type E. coli protein structure. And that helps us to gain insights as to which residues are most strongly impacted by trimethoprim treatment. But all of these insights still, remain high level, and of course, they've only been applied to the E. coli variant so far. But what we can also do is we can separate the data by each of the individual DHFR variants that we have in our library and determine which mutations infer resistance. And so, for example, here we're looking at Bacillus cereus. This is a pathogenic DHFR variant. And the red line here in the chart indicates the fitness response of the design variant. And then the blue lines, they represent the fitness response of mutant variants that do not appear to influence fitness to the point where it's greater than -1 at the high end of the trimethoprim gradient, right? And then the yellow line represents a mutant variant that clearly induced trimethoprim resistance across the experimental gradient. And from here, we can align these mutant sequences to the reference sequence and determine which amino acid substitutions at which position inferred resistance. And so in this case, with Bacillus cereus, it was a single amino acid substitution from valine to alanine at position 71. So we gained a new insight about which mutants led to resistance in this particular DHFR variant. We can also look at a more nuanced example. This is from Streptococcus pneumoniae. We recovered multiple different mutant variants that infer resistance to trimethoprim. And so from here, you can see that we have a range of amino acid substitutions across that protein sequence that infer resistance to the wild type. And so these include examples of single amino acid substitutions that infer resistance. There's even cases of 3 or 4 amino acid substitutions. And in this case, it was, the top line there in yellow is a 4 amino acid substitution that inferred the greatest fitness response for Streptococcus under the trimethoprim treatment. But it's also important to sort of keep in mind that we actually have this kind of data now for every single DHFR variant in our 1536 library. And we can evaluate these for the species-specific insights into antibiotic resistance within the DHFR protein family, right? And so this represents thousands of simultaneous insights into DHFR resistance across our entire trimethoprim gradient. And so together, these data, they represent basically a first of its kind study of variant effects at a scale not previously thought possible. Okay, where do we go from here? So future directions include coupling the results from our synthetic gene libraries with machine learning models to navigate fitness landscapes. So everything that I've sort of showed up to this point was data that we were able to pull out ourselves and sort of parse through and figure out what was going on. But by incorporating this into machine learning models, we're really hoping to unlock a lot more insights into the variant effects. And so for this process, basically, we would start with our sequence alignments and develop fitness landscape interaction models using probably a combination of supervised and unsupervised machine learning models. And then, we hope to gain useful insights, as I said, into these resistance pathways that these DHFR homologs that they can develop through these minor mutational changes. And these likely occur because of adaptive evolution under the different selective pressure from the antibiotic treatment. So I hope that I've sort of shown from this proof of concept study that Dropsynth can establish a foundation for next generation variant effect mapping, helping to enable this large scale exploration of how mutations shape antibiotic resistance, and then helping to predict resistance, before it emerges. And I believe it's these kinds of insights that are crucial for understanding how resistant pathways emerge through mutations. That helps to guide targeted drug design within the DHFR family. It's also possible to be informing broader strategies to predict and prevent antibiotic resistance in general. And so I just want to end by acknowledging all the members, past and current, from the Plesha Lab that have helped with this study, especially my mentor, Kaylin, along with Carmen and Sam. And then, of course, I'm happy to take any questions that you might have.
All right, thanks, Carl. That was a really exciting and comprehensive talk. Lots of very cool insights. So I encourage anybody to write your question in the Q&A box, and I can read it out to Carl to answer. I can also maybe start with a question or two. I'm wondering, I'm much more in the human gene kind of space and not in the bacterial space. I'm wondering how you're thinking about applying what you've learned through this single gene to other types of genes that have this kind of known involvement in antibiotic resistance, and whether you think there's any sort of unique observations you might make with different classes of genes, or if you think that there'll be a lot of broad kind of insights and similarities.
Yeah, no, it's a great question. And sort of one of the really exciting things that we found from this study is we can take this really diverse protein family for DHFR. And I think in the phylogenetic tree, the relatedness goes down to only 24 or 25% in some cases from the wild type E. coli. And we can still transform it into the E. coli knockout and have it actually perform its intended function. Right. So there's a lot of potential then to study pretty much any protein family using this approach where you can actually get them to work in knockout models in the lab. So it gets over this culturing sort of limitations. So I think it's really exciting. I think we could definitely apply it to many different targets for sure.